Protein's three-dimensional structure has a direct influence on its function. The structural-functional relationship is so tight that at times, even the change of one amino acid may produce a non-functional protein. For example, consider what happens in sickle cell anemia, a genetic disorder caused by the presence of abnormally structured hemoglobin. Hemoglobin is the protein in red blood cells that carries oxygen to the tissues through the bloodstream. In sickle cell anemia, a change in a single amino acid of hemoglobin causes the deformation of the red blood cells. Let's find out why. Hemoglobin has a quaternary structure, meaning it's composed by more polypeptides. There are 4 polypeptides, identical in pairs, which are called alpha chains and beta chains. Each chain has a central heme group, a molecule that can bind and release oxygen. Those with sickle cell anemia have hemoglobin with a different amino acid on the beta chains, shown here to view it closer. It involves the valine amino acid, which is located on the alpha helix close to the amino terminal end of the chain. In a normal protein, glutamic acid occupies this same position. The 2 amino acids differ in structure and chemical characteristics. Glutamic acid is negatively charged and behaves like an acid when in a solution. Conversely, the valine is a hydrophobic amino acid, meaning it tends to interact with molecules of the same type when in an aqueous environment. Substitution of glutamic acid with valine is due to a DNA error, a point mutation of a single base. Thymine instead of adenine results in a different codon, which leads to the addition of a different amino acid during protein synthesis. Let us compare the effect of one or the other amino acids in the 2 proteins, altered on the left and healthy on the right. Problems arise when the heme group in the middle of the chain releases oxygen and the hemoglobin changes shape. When the protein passes from the oxygenated to the deoxygenated form, the 2 subunits in fact rotate relative to one another. The mutation affects the way the chains are arranged. In mutated hemoglobin, the alpha helix where valine is located becomes more exposed. At this point, the valine interacts with a hydrophobic region of another molecule of hemoglobin. In this way, the 2 mutant proteins clump together. The process repeats and involves many molecules of hemoglobin, thus leading to the formation of fibers. Stretching the fibers causes deformation of the red blood cells, which takes on a sickle shape. They can even cause the rupture of the cell membrane, causing cell death. In any case, the sickle shape makes red blood cells more fragile and shortens their lives. The result is a reduction in the number of red blood cells. When the number of red blood cells drops below a certain level, the result is anemia, a condition linked to various diseases and a lowered capacity of blood to carry oxygen. Those with sickle cell anemia have these typical symptoms: fatigue and shortness of breath. Other symptoms are sudden pain, thrombosis, and infection. Some symptoms depend on the sickle shape of the red blood cells. We have seen that erythrocytes take on a sickle shape when hemoglobin gives oxygen. This happens in the capillaries, the smallest vessels of the circulatory system. The sickle shape can cause red blood cells to adhere to capillary walls, resulting in microcirculatory blockage. The severity of sickle cell anemia depends on how much abnormal hemoglobin is present. We have seen that this alteration is caused by substituting one amino acid and upstream in the corresponding gene by replacing a single nitrogenous base in the DNA.